Welcome everyone to the IIVS-sponsored webinar on the Reconstructed Human Skin Micronucleus Assay, a novel micronucleus assay in a 3D human skin model. This webinar is provided as part of IIVS other than the panelists has been muted and will remain so for the duration of the webinar due to the large number of attendees. You may ask questions by using the question feature at the bottom of the control panel to the right. Click on the arrow to maximize the control panel and view the questions window. Questions can be submitted anytime. They will be answered during the last 10 minutes of the webinar. And we are recording this webinar and the question and answer session at the end. So any questions that are not able to be answered within the scheduled webinar time will receive a written response that will be distributed to all webinar participants, along with a PDF of the presented slides and a link to view the webinar recording. Let's meet our speakers. Today's first presenter is Greg Munn, Study Director at IIVS. Greg serves as the lead scientist for IIVS's work on the RSMN assay and has trained both internal staff and external scientists on the technical performance of the assay. Our guest speaker and second presenter today is Dr. Stefan Fuller, global expert on genetic toxicology at Procter & Gamble. Stefan serves as the chairman of the Genotoxicity Expert Team for Cosmetics Europe, the Personal Care Association. Now that you know a bit about the speakers, I will pass the presentation controls over to Greg so that he may begin. Give me one moment here.
Show my screen?
Yep, you're good.
Thank you, Mandy, and thank you for all participants to this IIVS sponsor webinar. My name is Greg Munn. Again, I'm one of the study directors at IIVS responsible for the Reconstructed Skin Micronucleus assay refinement and studies. I'd like to start my presentation with a brief overview of cosmetics genotoxicity testing procedure, followed by mentioning some of the milestones achieved in first developing the RSMA and then assessing the performance of the RSMA. And finally, although this is still work in progress, development of the automated scoring process for the RSMA that can certainly introduce a significant efficiency to the existing assay system. Again, my presentation will follow by the review of the Cosmetics Europe's in vitro genotoxicity pre-validation program, and that portion of the webinar will be presented by Dr. Stefan Fuller of the Procter & Gamble. In a typical genotoxicity procedure for cosmetics ingredient, one would typically start with the quantitative assessment of the chemical. That means gathering the existing knowledge for its safety assessment. If that information is considered insufficient, you can employ some basic in vitro assays. Examples are reverse mutation assay, AIMS, mouse lymphoma, chromosomal aberration testing, and in some cases, photogenotoxicity testing. One concern is that there is a high rate of false positives, especially when you combine two or more of these in vitro assays. That may result in false positive rate of approximately 90%. In the past, The next tier testing method was employing the in vivo testing, such as bone marrow, unscheduled DNA synthesis, or even rodent skin micronucleus assay. But I'm sure, as we are all aware, as of March 2009, the European Union's 7th Amendment to the Cosmetics Directive makes the in vivo genotoxicity testing for cosmetic ingredients impossible. Because it plays in vivo genotoxicity assays played an important role, that really identified the need to develop an in vitro test, certainly that is more relevant to cosmetics testing, that is perhaps a test system based on skin, and one with the better overall predictability than the current battery of existing in vitro assays. One of the proposals was the development of a novel assay called the Reconstructed skin micronucleus assay. This initial inquiry was spearheaded by Dr. Marilyn Artema, then at Procter & Gamble. IIBS was engaged to initiate the development of the assay using the epiderm tissue. Epiderm tissue is a three-dimensional tissue structure manufactured by MatTech Corporation. Some good aspects or some good characteristics of the epiderm tissue was that, in fact, cells are human in origin. This is a three-dimensional tissue structure similar to human epidermis, and it has a presence of stratum corneum, which mimics the outer barrier. First, a bit of a story about the epiderm-based assay development. In this slide, you can see a picture of the epiderm tissues on the bottom right-hand side. Cells are grown on a cell culture. Epiderm is located on the bottom surface, which is the collagen-coated porous membrane. And if you look at a mature epiderm model, or a model that is ready to be used in an assay, you can basically see that it's about 8 to 12 cell layers thick. On the bottom, you have a well-organized or active basal cells. But as cells migrate upwards or pushed upwards to the outer surface, you have a different degree of keratinization, ending up as a stratum corneum, again mimicking the outer barrier. Now, unlike the monolayer cell culture, this really represents the full spectrum of cell differentiation or keratinization. So you can certainly count on multiple cell conditions that's much more difficult to evaluate compared to the monolayer cells. For the micronucleus assay, we use the 6-well trace, left-hand side. Cell culture inserts are placed into each of the 6 wells, aliquoted 1 ml of new maintenance media that feeds the cells from underneath and maintains the tissue structure. Is aliquoted onto the bottom of the well, and again the inserts are placed directly on top of that. The test chemical can then directly apply to the dry topical surface of the tissue, as you can see in the picture. Now, when we initiated the development of the epiderm-based micronucleus assay, we didn't have to start from scratch. There was a similar protocol for rodent skin micronucleus assay published by Nishikawa, and we based our micronucleus assay based on that. By that, what I mean is the dose volume per surface area is similar to that. And in the epiderm-based micronucleus assay, you also have a repeat exposure process. Two main technical challenges really had to be addressed. Again, because the epiderm tissue covers cells at multiple stages of differentiation, one had to really identify the cells that are actively dividing. And one solution to that is the use of the cytokinin B, which is a cytokinesis blocker. and you have to optimize the exposure process and concentration of the cytokeratin B. The second significant item that had to be developed was method of trypsinizing or isolating the single cell suspension from the epidermal tissue structure. And this is an example of a typical in vitro micronucleus assay. For a viable cell, you have a chemical treatment, and if in fact the chemical is genotoxic, perhaps you have a development of the micronucleus. And in absence of the cell cytokeratin B, cells would continue its division, and perhaps you end up with two daughter cells in this example, one containing the micronucleus. Again, because it was essential in the epiderm-based micronucleus assay system to identify the actively dividing cell, you incorporated the cytokeratin B, and in the assay, along with the chemical treatment, you have a cytokeratin B exposure through the media. Again, cells would continue its dividing process, but rather than ending up with two daughter cells, you have an active cell that divided, ending up as a binucleated cells. And if there was a genotoxic event, binucleated cells with a micronucleus. So this optimal procedure of cytokeratin B exposure really allows you to identify the proper population of cells within the epidermal tissue structure and allow you to do a proper cell count or micronucleus event count in binucleated cells. The second item that I had mentioned was developing the proper trypsinization process. I think the actual steps are mentioned in numerous publications, so I will just go ahead and show you the beginning and the end result of the trypsinization. You can start with the epidermal tissue, and by employing this modified trypsinization process, you'll end up with a tissue pad or the remains of the stratum corneum, and perhaps one or two layers of highly characterized cells. And the cells that are necessary for the micronucleus evaluation, perhaps the basal cells and suprabasal cells, can then be collected, fixed, and placed onto the slide. And on the bottom example, you have that particular cell population stained with the acrylic in orange. Our current experimental process is the 48-hour exposure cycle. If you look at the timeline, on Tuesday, we would get the tissues from Attay Corporation. Typically, the tissues are transferred from the shipping tray into the 6-well and incubated overnight. On Wednesday, we would start the dosing process. That means replacing the medium underneath with the fresh medium containing cytokeratin B, and topically dosing 10 microliters of the chemical directly onto the surface of the epidermal tissue. Approximately 24 hours later, on Thursday, that dosing and repeating process would be repeated. Again, a fresh media containing cytokeratin B would be added to the bottom of the well, and the top 10 microliters of chemical would be applied topically. On Friday, which is approximately 48 hours following the initial dose on Wednesday, the epidermal tissues would be collected and trypsinized to generate the single-cell suspensions, which will then be fixed and placed on slides and counted for or scored for the micronuclei. Our basic procedure is that, in fact, initially 500 cells would be counted for the percent binucleation rate, and from that you can get the percent survival. In the beginning, one of the criteria for the negative control would be it had to have at least 25% binucleated cells that ensured that the tissue conditions were proper. Tissues with too much cytotoxicity or significant cytotoxicity of 60% would not be scored for micronuclei. Again, typically 1,000 binucleated cells per tissue are scored or counted for the presence of the micronucleus. Now, based on this initial development of the basic assay procedure, we wanted to go ahead and assess the performance of the assay. And these are some of the basic questions that we had asked. Can stable background be established, or stable negative and positive control be established? And can we identify the proper positive control? Can the assay procedure be transferred to multiple laboratories and generate reproducible or similar results among many labs? And can it correctly identify true genotoxins and true non-genotoxins? And finally, for the RSMA, can it identify genotoxins that require metabolic activation? Again, the basic procedures for the assay was developed by IAVS and then successfully transferred to 2 additional labs in the beginning, the one at Procter & Gamble and at MatTech. The initial indications were that the assay procedure were readily transferable and the reproducibility among labs were good, as shown by this example of the negative control. All of the labs were able to generate stable negative control percent binucleation response ranging from 40 to 47%. And the micronucleus event for this negative control was stabilized at approximately 0.1%. Mitomycin C was at early stage identified as the usable positive control, and at ranges of 3 to 10 micrograms per mil prepared in acetone, it provided a statistically significant response in the epiderm tissues. That was done according to the Fisher's exact test single tail. And just to go over some of the values that we collect from the assay is are the percent survival that's based on percent binucleation of the tissues treated with chemical compared to that of the negative control tissues. Typically, 1,000 binucleated cells are counted for the micronucleus event. And although we mentioned the 25% as the acceptance criteria for the percent binucleation of the negative control, we were typically able to achieve approximately 40 to 50% binucleation rate with a stable, very low micronucleus frequency of approximately 0.1%. Now, no true known genotoxins and perhaps 4 non-genotoxins in the rodent skin micronucleus assay were tested among all 3 labs. In this slide, mitomycin C, vinblastine, and methylmethane sulfonate, which were all true genotoxins. Had produced stable positive and negative responses among all 3 labs. Finally, one of the other assessment point was can the epiderm-based micronucleus assay identify genotoxins that require metabolic activation. Some of the gene profiling work was done in parallel. I won't go into detail. Again, these are all in publications. Indicated that, in fact, there were a lot of similarities between the epiderm tissue compared to the normal human skin. That led to indication that, in fact, the epiderm tissues may be metabolically competent compared to the human skin. However, one of the chemicals that was tested at IIVS, cyclophosphamide, is a true genotoxin that requires metabolic activation, showed a steady negative response in the 48-hour exposure protocol. One of the modification was proposed and tested, which was expanding the exposure time from 48 to 72 hours. Again, this is the timeline. We would get the tissues on Tuesday as usual, but rather than putting the tissues for overnight incubation, tissues would be incubated for approximately at least 1 hour, and we would start the dosing process at that point. That means, again, adding the media containing cytoclasm B to the 6-well and applying 10 microliters of test chemical directly onto the topical surface. The dosing and repeating process would be repeated on Wednesday or approximately 24 hours later. Again, on Thursday, approximately 48 hours later. And on 72 hours following the initial dosing, tissues would be harvested and fixed and placed on the slide and counted for the micronucleus.